Friends, I'm going to talk about the epidemiology of sickle cell anemia. Sickle cell anemia is a disorder related to the blood cells. Sickle cell anemia gives sickle shape to the RBCs during hypoxia. That means during less oxygenated conditions, you can see the formation of the sickle cells. Han and Gillespie 1927 identified the sickle cells in hypoxic condition. See, the normal RBC, normal red blood cell, is looking like a perfect donut, and sickle shaped you can see for the sickle red blood cells. So this is not new. This has been reported by the Hippocrates 460 to 430, 337, 377 BC. So another sickness of the spleen. He narrated this as a problem of the spleen. It becomes on mainly in the springtime. That means he correlated the disease with the season. Now also you can find some of the symptoms or the severity of the disease during the spring season, and this is caused by the blood. That means spleen. So splenomegaly occurring, and the sickle cell anemia, the severity or the vascular crisis or the problems are related to the season also. The Hippocrates has defined at the time itself. So the spleen becomes enlarged with the blood. Yes, this is called splenomegaly, which evacuates into the stomach. Shooting pain, shooting pain in the spleen, breast, the clavicle, the shoulder, and beneath the shoulder blade. So all these things, pain is the characteristic feature of the sickle cell anemia, and splenomegaly can be seen. That means hemolysis is occurring in this one. This has been noted by Hippocrates in 460 to 377 BC. So how the sickle hemoglobin is formed? As you are all aware that sickle cell anemia is a genetic disorder. This occurs because of a change in the nucleotide. So the amino acid that is coding the sixth codon amino acid, that is glutamate. Glutamate is a normal highly negatively charged amino acid. The change of the middle nucleotide A to T, that means GAG to GTG, leads to change in the amino acid, that is valine. So this valine is the non-polar. So because of the glutamate highly negative charge, and because of the valine it is non-polar, or the near neutral charge will be there. This can be seen in the entire hemoglobin. So the change of the glutamate to valine, that changes the charge of the hemoglobin, total hemoglobin. If it is glutamic acid, if it is highly negative charge; if it is valine, less negative charge. That means sickle protein is having less negative charge, and normal protein is having more negative charge. So this is a hypothetical picture I have shown you. I have shown both mother and father, father and mother, father and mother, father and mother. All I have taken just as a heterozygous condition. They are the trait or the carriers. They are heterozygous for they are having one copy of the normal hemoglobin and the other copy of the sickle hemoglobin. Both, all the mother and fathers are like that only. If mother and father both contributes to normal hemoglobin, then the person sibling will be the absolutely normal. And whereas if father or mother contributes one defective and one normal, that becomes a trait or a carrier. He is absolutely carrier. He will not develop any symptoms of the sickle cell anemia. Both mother and father contributes the defective allele. That means defective hemoglobin to the sibling. He will become the sickle cell anemic individual. So that means, so from this it is clearly known that it is an autosomal recessive blood disorder. That means due to the inheritance, you can find if two heterozygous individuals or the parents are mating, approximately 25% of the individual SIBs will get the disease, 50% will become carriers and 25% will be the normal. This is the ratio. This is the population level. So here you can see the pictures: oxyhemoglobin A, oxyhemoglobin sickle. So this is the normal oxyhemoglobin with oxygen, normal hemoglobin with oxygen, sickle hemoglobin. So this notch is an hydrophobic end that is generated because of the valine at sixth amino acid, this notch. So whereas after, whether it is a oxyhemoglobin or the sickle hemoglobin, after releasing the oxygen in the tissues cells, so it will develop, it will have one 85 to 87 amino acid inherent notch, and sickle hemoglobin also will have both the things. So this is the normal 85-87, and sickle hemoglobin, this is of 6th amino acid. So you can find this type of things in deoxygenated condition. When oxygen deprived condition or the less oxygen condition in the tissues, you can find this one hemoglobin's valine. You can see this one hemoglobin's valine is attached to the 85-87 of the other hemoglobin or adjacent hemoglobin. You can see this is the formation of the strand, polymerization that is occurring in the sickle hemoglobin. So this is one hemoglobin at 6th amino acid. This is the other hemoglobin, that is 85-88 amino acid position. So you can see the attachment. So like this, one hemoglobin, sickle hemoglobin, you can find the polymerization. During the polymerization process, you can find the participation of 7 pairs of hemoglobin molecules, hemoglobin chains. So here you can see that 14 hemoglobin, that is 7 pairs of hemoglobin is participating in the this one. So X-ray diffraction studies clearly showed that these many molecules are participating in the polymerization process. Because of this only, it becomes very stringent polymerizing substance. So it even changes the structure of the RBC. So if it is normal hemoglobin, there will not be any additional hydrophobic end. It will not merge with the neighboring hemoglobins, and it is freely flowing in the blood vessels, and it will not cause any obstruction in the capillaries. You can see free flowing. So whereas the presence of sickle mutation, that is GTG, so additional hydrophobic end is formed. It is adjacent hemoglobin's 85-88 amino acid, it is attaching, it is forming the polymerization. The clumped hemoglobin is changing the sickle shape, donut shape to the sickle shape, and it clogs or obstruct the small capillaries, and rear part will not receive the blood, and we can find the ischemic conditions in this one, and all sorts of pain and other clinical manifestations can be seen because of this obstruction. So the difference between the normal and sickle shaped cells is, the normal cells is a disc shaped cells. These are all deformable. Once it releases, again it oxygen, and again it reoxygenates, and again its lifespan is approximately 120 degrees centigrade. They are almost round and donut shaped. Whereas in the sickle cell anemia individual, the cells are sickle shaped. Some of the cells you can see that sickle shaped cells is there, and it is very rigid because 14 strands are participating in the polymerization. It's very rigid sickle cell molecule. Even some few instances it will go to the deoxygenated condition, and again it forms to the, now it comes to the normal shape, all the things, and it can live only approximately 20 days or less than that. It depends on the body, other hydration levels, other conditions, depends. However, it can stay approximately 20 days. So here oxygenated condition and deoxygenated condition, again it will come back to the normal oxygenated condition. So whereas the sickle cells, HbS containing cell, it goes to the normal and it will not come to the normal, but this will not happen sometimes. So it will come to the it will be staying in the same, so it will be very rigid. That's why it is, even if you are giving oxygen also, it will not come to the normal donut shape. It will be staying in the same stays. So like this, this happened because of this. The physiology, pathophysiological changes can be seen. So this is the beta globin sixth codon, and if it is normal, the glutamic acid, it is a normal and it's freely flowing. Whereas GTZ, that is the valid nucleotide, here you can see that polymerization formation, and here the normal hemoglobin is there, and the hemoglobin is changed to the sickle hemoglobin. It obstructs the small capillary. The entire sickle cell anemia is shown in this picture. So deoxygenated conditions, polymerization of hemoglobin can be seen, sickling of red blood cells, endothelial damage and activation can be seen, RBC and leukocyte adhesion to the endothelium and vasoconstriction can be seen, vascular occlusion, organ ischemia, and end organ damage can be. This is the essence of the pathophysiology of sickle cell anemia. So pathophysiology of sickle cell anemia, if you see, it affects the hemoglobin. It affects the hemoglobin. Hemoglobin will not come to the normal stage. So polymerization of the hemoglobin leads to the cascade of effects, decreasing the blood flow. So as it is clogging the small capillaries, you can see that obstruction of the blood flow, and tissue hypoxia causes the acute and chronic damage. This can be seen in this individual. So different sickle cell disease genotypes and its prevalences can be seen in the population. Sickle cell anemia SS, HbS, that is a homozygous condition, it is 65%. HbC disease, this is particularly present in African countries, is approximately 25%. Sickle S beta plus, this is S beta plus thalassemia. That means on one chromosome sickle cell mutation, the other chromosome you can see that beta thalassemia mutation. So because of this, the synthesis of hemoglobin will be hampered, and whatever that is synthesized, some part will be the sickle hemoglobin also. Here this type of condition approximately 8% can be seen. Sickle beta 0, SG beta 0 thalassemia, it is very less. That means they are thalassemia major and also sickle cell disease can be seen in this individual. So this is about the prevalence of different hemoglobin variants. So last lecture also I told that the region specific or discontinued distribution of different hemoglobin variants. So sickle hemoglobin can be seen in the central India and many parts of the Africa. Here in the central India, you can find in Madhya Pradesh, Gujarat, Maharashtra, Chhattisgarh, and some parts of Tamil Nadu and Kerala regions. So the hemoglobin C is exclusively present in Africa. Very small portion of the region is having that. And HBD is located only in Punjab. HBE is present in West Bengal and Southeast Asian countries. Alpha thalassemia occurs in all these regions as well, because alpha thalassemia some extent gives the protection. So here you can see the sickle cell as a multi-system disorder. So it is not affecting one organ, even though the cause for the disease is one single mutation is causing the disease, but it affects almost all organs of our body. So first one is the cerebral, ophthalmic, cardiovascular, respiratory, gastrointestinal, musculoskeletal, and genitourinary systems. So almost all the systems are affected. So who is affected? So mostly descendants of Africa, tropical parts of Africa, eastern parts of Niger, river around the North Ghana, Benin, Central African Republic regions. You can find in Asia, you can see Burma, Thailand, Laos, Cambodia, Malaysia, Indonesia, and India. You can find in that regions. In America is largely populated African American areas. Wherever it is Los Angeles, you can find that region, you can see that. So it is affected in Americans, means American black Americans, not the white Americans. So you can see that. So the geographic distribution of the sickle cell trait and malaria is strikingly similar. So that means, so it gives the scope for us to think about the disease, that the disease emerged in the population as a protection against the malaria. So for this hypothesis, you can see that pre-interventional malaria maps and sickle cell anemia frequency, allele frequency, are strikingly similar in the world. It has been superimposed and studied, and this. So the distribution of HbS in 1961, you can see this. So here you cannot find that Central India has not been screened at that time, and now some of the data is available, and many people are working in the Central India on sickle cell disease. So the distribution of HbS, that means beta sickle gene in Africa, is within Africa, the frequency of sickle beta and and accordingly the SS is highest in the low altitude equatorial regions. Low altitude equatorial region, you can find it more. 1 in 4 in Western Africa, 1 in 10 in Afro Caribbean regions. So that means the sickle allele frequency or the distribution varies from region to region, even within the country. So the prevalence of HbSS by age, these are the three different studies were superimposed here, and these studies shows that the children surviving before 5 years or more. So after 5 years, this one after infancy, the survival is very less. That indicating that excess of mortality in individuals with sickle cell anemia. The HbS gene frequency in different areas of Saudi also, you can see that even one part of the Saudi, the frequency or the clinical manifestations are entirely different from the other parts of the Saudi Arabia. So the first case was reported in the eastern province in 1963. In Saudi, it is identified only in 1963. HbS gene was encountered in all areas screened in the kingdom. So HbS gene frequency was highest in the eastern province of Saudi Arabia, followed by the southwestern provinces. So approximately 23.98 million population of Saudis are affected, are present, and the carrier status is 2 to 27%. Different regions, different frequencies is there. And SCD prevalence is 1.4%. Out of total 23.98 population, 1.4% of the individuals are having the sickle cell anemia. The clinical phenotype has two major forms. So eastern side and western side, you can see the clinical differences. So the haplotype of the eastern individuals is, eastern population Saudi Arabia is Arabic Indian, and western part is the benign. So if you see the acute chest syndrome, it is very less in the eastern part, and recurrent acute chest syndrome is very less, dactylitis is less, painful crisis is late, and stroke is less, silent vein infarcts are late, and body build is normal. So this type of clinical conditions you can see the eastern region, whereas in the benign haplotype, the western region is comprising, and you can see the clinical severity is severe, acute chest syndrome is more, recurrent acute chest syndrome is more, dactylitis is more, painful crisis you can see in the very early, and stroke is more, and silent brain infarcts also very early you can see, and the body build is also less. A low body build you can see. The eastern parts have more deletional alpha thalassemia. The main difference why the phenotype or the clinical manifestations are not that much severe in eastern part is, eastern part individuals are having more of deletional alpha thalassemias, which gives protection for the individuals. That means presence of alpha thalassemia with a sickle cell anemia will give the protection. And higher total fetal hemoglobin levels and higher total hemoglobin and fetal hemoglobin can be seen in this group of individual when compared to the western part of Saudi Arabia individuals. Eastern part is slightly milder than the western part. The prevalence of SCD in USA. As I told earlier, the sickle cell disease in USA populations affects approximately 90,000 to 1 lakh Americans. That too, only the native Americans is very less. Only black people are having the much of the sickle cell anemia. So black Americans are having the hemoglobin, this particular hemoglobin, that is sickle cell anemia. So sickle cell anemia in UK, if you see that approximately 3 lakhs to 5 lakhs births per year in UK, 3 20 SCD births per year in UK, and 50,000 affected individuals you can see, and three-fourths live in the London. London is having more of the sickle cell anemia patient when compared to the rest of the UK. So in India, the presence of sickle allele is present in the tribal groups of Nilgiri Hills. In 1952, Lehman and Cutbush first time identified in 1952 in Nilgiri Hills. In India, incidence and prevalence of SCD very high, especially in the central and western regions. Even though the reliable epidemiological data are not available with a population of 1.25 billion individuals, India is estimated to be home for over 50% of the global SCD population. So India is the home for the more than 50% of the global SCD populations. So that much severity is there, that much risk is there in India because of the sickle cell anemia. So sickle cell distribution, if you see that HbS allele is mainly concentrated in the scheduled tribes and scheduled caste and other backward caste populations of Madhya Pradesh, Orissa, Chhattisgarh, Jharkhand and Gujarat. So this particular belt, it is there. Andhra state, you can see some regions of Andhra state and Kerala and Tamil Nadu region also. You can find in Tamil Nadu region there some of the tribes are having this particular thing. So the frequency ranges from 5 to 40. It is not uniform. In some places it is very less, in some places it is very high. And central India it is more than when compared with other parts of the. However, no attempts have been made so far to address the issues related to the disease so far at the national level. So even though this much burden is there, the government of India has not taken much initiative to solve the burden, to reduce the burden of the sickle cell disease in India. Thank you very much. If you like this lecture, please share to others. Thank you.